Hello, we are team number 5 and this is our third lab of biology. This lab is about micropropagation of seeds and we had a question, which is how is the development and growth of a plant affected when it is in different light conditions? Well, agriculture is a fundamental sector of biotechnology. It consists of a set of activities and techniques carried out by humans to cultivate the land and obtain goods or products. Generally, foodstuffs and textile fibers. Well, these products are obtained from the standardized treatment and care of plants, whether vegetables or fruits. These are used to meet one of the most basic human needs, such as food. Agricultural techniques can vary according to the product to be obtained, and one of the most sophisticated techniques used in agriculture is micropropagation. Well, in micropropagation, this is a technique used in biotechnology for cultivation of plant tissues with the purpose of reproducing them in a controlled manner in a laboratory, using an optimal medium for further growth. Basically, we provide with nutrients necessary for the tissues of plants to survive and reproduce in the place where it is located. Because micropropagation techniques are developed in a laboratory, it is common that the researcher working with them must take into account conditions in which they must be, such as temperature, light, pH, salinity, among other factors. All these factors can affect a plant growth and are essential to maintain optimal conditions for successful micropropagation.
Continuing what Alan said, in the experiment we located 6 different seeds. 3 seeds of cucumber and another 3 seeds of radish. These seeds were located inside a plastic flask and we used a technique commonly used for cultivating seeds. The technique is called in vitro. This technique consists in having, which is defined as cultivating plants inside a flask with artificial conditions. That means without dirt, without water, etc. Just a artificial gel that is called moulissages Skov. This is commonly used in laboratories in order to grow plants. The nutrients that are located on this gel are high levels of nitrates, as well as many additives like sugar, agar, vitamins, and growth regulators. For this laboratory, it is needed to have no fungus inside the flask. That's the reason why if you unzip the flask, a lot of bacteria can come in, or that's the reason that we use latex gloves in order to not contaminate inside the flask or contaminate the seeds. We also use sterile forceps for the sterile situation and a located candle which make possible a zone of fertility within 15 centimeters around. To conclude the theoretical framework, we expected to make a plant grow in artificial conditions without developing any fungus with the process in vitro and with the artificial gel called moulissages Skov. In the flask, we mark a line with a Sharpie in order to see how many centimeters the plant grows after some days.
Well, now we're going to move to the methodology. The dependent variable is the plant growth. The independent variable is the in vitro process, and the control variable is the seed of cucumber and seeds of radish. Materials we used were 2 flasks with the culture medium for plants, the sterile cucumber and radish seeds, 1 candle, 1 lighter, and 1 sterile forceps, and a ruler and a pen. Now I will be narrating the process of our experiment. We will start with the methodology. First of all, light up the candle. Now we started taking the seeds with the sterile forceps. We bore them a little bit for making the steriles and place them on their respective flasks with the culture medium for plants. We can see here our friend Solano making as well. We mark them with a Sharpie for then looking how much they grew. It was time for leaving them on a place with no light. We choose the locker. After 2 days, we look at the process of growth. The radish is already germinated, while the other one didn't. Finally, 4 days after the last video, we look at them and we saw that the radish seed was 16 centimeters higher, but the radish one didn't germinated yet.
So I'm going to analyze the results of this project. We can see that the radish seed grew 16 centimeters in 6 days, and it had no sunlight. You are able to see in the images how much did the seed grow. This happened because of the Murashige scoop gel that was located in the seed. These gels gave the seed all the nutrients that it needed to grow and germinate. The reason why the radish grew and the cucumber did not is because the radish takes approximately 3 to 5 days to germinate, while the cucumber takes approximately 7 to 10 days to germinate.
To conclude this project, the Murashige scoop gel helps a lot in the process of agriculture and micropropagation of seeds. This process of cultivation, or in vitro, which we already talked about previously, makes it very easy for all the processes of germination to happen, which is very useful because it is not required to have all the needs that a regular plant uses, like it is dirt, water, and sunlight. And with this, it is very possible to have a big future on micropropagation of seeds, which may generate less contamination and help the growth. So, the growth of seeds are affected by the situation where they are located and the environment they live in. Thanks for your attention and see you next semester. Listo.